Good morning, everyone. Let's get started. So today we will finish the lecture one about the cardiovascular system. But before we dive into the PowerPoint slides, let's do a quick review. If you could please pull out the study guide and turn to page 2. We are going to label some of these diagrams as part of our review. Okay, so this diagram over here, label the chambers of the heart, use blue and red to indicate oxygen level. So let's see. Over here, we have the top right chamber, and that would be the right atrium. And right atrium is going to be poor in oxygen, so I'm going to use blue. Below the right atrium, we have the right ventricle. And that is also going to be low in oxygen. On the other side, we have the oxygen-rich blood going through the left atrium. And of course, below the left atrium is the left ventricle. Okay, so this is again O2 rich, and on the other side is O2 poor. Separating them, of course, is this wall, which you should know by now is called the septum. All right, so nothing's new, just things that we talked about last time. Over here, number 5, label the structures leading to and away from the heart. So we have various vessels here. So starting at the top here, we have the biggest vein, right? At the top and at the bottom, of course. Those are our superior vena cava, as well as the inferior vena cava, collecting blood from the upper part of the body as well as lower part of the body. And of course, they drain into the right ventricle. Over here, we have another vessel. I'm just going to use a slightly different shade of blue, like that. That would be, sorry, just going to find the same color as before. That would be the pulmonary artery. Pulmonary artery, taking the blood to our lungs, right, for reoxygenation. This is part of your pulmonary circuit. And then the blood will come back through the pulmonary veins. On the left side as well as the right side. Okay. And finally, we have another big vessel here. Again, I'm just going to use a slightly different red. And that, of course, is the aorta, right? The biggest vessel in the body. Aorta. Now, I told you the aorta arches behind the heart and continues downward, right? If you look at the 3D model on eCentennial, you can see that this right here, that's the aorta. And if I rotate the heart, you can see it arches back like that, right? It continues bending downward behind the heart, and you know, you can see it emerging down here below in our diagram. Right? So over here, I can add a little bit of a vessel here, right? That is also going to be our aorta, but it has a different name now. Once it comes down here, and we wrote it down last time, I hope you remember what it's called. That would be the abdominal Abdominal aorta. Are you guys okay? Anybody have any questions so far before I move on to the next one? All right. Label the 4 valves. The purpose of valves is to prevent backflow of blood. We don't want things to be moving backwards. But you know, if you have a leaky valve, for example, regurgitation can occur and blood might be squirting backwards, which you know could lead to things like heart murmur. We talked about that last class as well. But generally speaking, in a normal, healthy heart, we should not have any backflow, thanks to the 4 valves that we have. So we have a valve right here separating the right atrium from the right ventricle, that valve. Anybody remember the name of that valve?
Tricuspid.
Thank you. That is the tricuspid valve. 3 flaps. And on the other side, we have another one right here. It's a little bit unclear, this diagram, but there is the bicuspid valve, right? And together they open and closes with each other, right? Always in sync. That is the AV valve. On the other hand, you would have the pulmonary valve right here. And the aortic valve down here. Aortic valve. And those would be your semilunar, half moon looking valves. Okay, semilunar will also open and closes together, but out of sync with the AV valves. All right. So just very briefly, you can see again on this diagram, that's the tricuspid valve right there. And on the other side, we have the, it's called a mitral valve or the bicuspid valve, right? Both are acceptable. I just tend to stick with the bicuspid because it's easier to remember, right? If I look at the heart from the front like that, I remember the number like 32, right? 32, three flaps and 2 flaps. So tricuspid and bicuspid, okay? So over here, we have the aortic valve going into the aorta, but you can see that the pulmonary artery has been cut away. Right? Normally in the diagrams that we are used to seeing, this would come right down here to connect to the right atrium, right ventricle, right? But they have cut that away, and as a result, from this model, you're not able to see the pulmonary valve. But of course, is there, right? Okay. So play around with this 3D model, right? You can actually click on the label here, and it will give you like more information if you need to read more about it. Okay, this is of course on eCentennial. Under study resources, 3D models. Okay. All right, finally, starting with the vena cava, trace out path of blood flow in the heart. So the blood's going to come in from the superior inferior vena cava, and it's going to go right over here. Okay, we just got to put 1 here. Going to go through the tricuspid valve into the right ventricle, and it's going to go into the pulmonary artery. Right, taking it to the lungs, where the blood will get reoxygenated, and they will come back through these number 4. Okay, those would be your pulmonary veins into the left atrium, down into the left ventricle, and then finally squeezing the blood throughout the body from the aorta. Okay. Now, because the okay, so let's talk about a little bit about the right ventricle versus the left ventricle. Which one do you expect to have a thicker myocardium? Myocardium is the muscle, right? Which one do you expect to have a thicker layer of myocardium, or do you expect them to be the same? The right ventricle, the left ventricle. What do you guys think?
Left ventricle.
Yeah, why is that, Shelley?
Because I just see the 3D model and then it looks thicker.
Oh, okay, yeah, that's a good way of telling. Very good. And you're correct. Okay, the left ventricle will be thicker, and here is the reason. Okay, let me write this down for you guys first. This has the thickest myocardium. Okay, the left ventricle. Okay, and the reason is we have to, well, we as in like the heart, the ventricle has to squeeze hard enough to generate enough pressure for the blood to go through the aorta and then get distributed throughout the entire body. Okay, so the entire body is really big, right? You can't just give it like a tiny squeeze. You have to squeeze it really, really hard to generate enough force to achieve that. On the other side, the right ventricle, because the lungs are just like very close by the heart, right? The heart is in the middle and then the lungs are on the side. Like you don't need to have a too big of a squeeze. You can just squeeze a little bit and then you will be able to arrive at the lungs and then the blood will return, right? So for that reason, the myocardium, the muscle, is much thicker on the left side compared to the right side. In fact, it's the thickest of all chambers. The myocardiums of the atria on the top, they don't need to create too big of a squeeze. Okay, they just need to have a tiny nudge, and then the blood will fall down because of gravity, right? Okay, so the squeeze is more for emptying the entire chamber. Okay, gravity is just going to take the blood down to the ventricles anyway, but by squeezing a little bit, it makes sure the chambers are going to be completely emptied. Okay, so if I ask you on the test which which chambers has the thickest myocardium, make sure you choose left ventricle. Is that clear? All right, I think that's as much review as we will do for now. If we have time, we'll take up more questions later. But I think it's good to go back to the lecture now. Give me a second. I think I lost my file here. Just gonna pull it up. There we go. Where did we stop last time? I think it was a conduction system, right? Over here, yep. Okay, actually, you know what? I lied. Let's go back to this study guide and write a little bit more. So we were talking about this conduction system last time before we ended the lesson. I just want to add a little bit more detail to it so you fully understand what's going on. So here, second part here, describe the series of events that takes place as the electrical signal passes through different parts of the conduction system. So at the beginning, right, the parts of the heart that generates the rhythm for you, that is a cluster of cells called the SA node, right? We know that already. Okay. Like I said last time, we cannot voluntarily control it, but then we can influence it through physical activity, consumption of things like caffeine, or certain medication would speed it up or slow it down. And then as it sends out the signal, some of the signal will spread across atria. Okay, and then some will travel towards the AV node. So as the signal spreads across the atria, and as the signal goes down to the AV node, a bunch of things will happen simultaneously. Okay, so we'll write them down. After the signal reaches the atria, or as it's passing through the atria contracts. Okay, so this is systole, right? what happens when the atria squeeze? When the atria squeeze, the AV valves open, right? You must open the AV valves for the blood to go down to the ventricles, right? So AV valve opens, and then the ventricles, so atria contract, that's a plural. Ventricles relax and fill up with blood. Okay, of course, the blood is coming from the atria above. If you remember what I said about the valves, if you open the AV valves, then you must close the semilunar valves, right? So all these things are happening as the signal is spreading across the atria. The ventricle, since it's relaxing, we call that diastole, right? All right. So after the signal gets to the AV node, it would then get sent to the Purkinje fiber, which are fibers that are embedded within the septum as well as the walls of the ventricle, right? I'm just gonna quickly show you, right? All these things. These are the Purkinje fibers. So the signal, once it gets to there, it will cause the ventricle to squeeze from bottom and up, so that the blood would squirt upward, right? So over here, once it gets to the Purkinje fibers, the following will happen. I'm just gonna use a different color to separate the events for you. So now the atria relax and fill up with blood. Okay, so superior and inferior vena cava supply blood to right atrium, and then pulmonary vein supply blood to left atrium, right? As the atria is being filled up, you don't want them to, continue to fall down to the ventricle. So it's important to close the AV valve. AV valves close. Okay. And since the signal is now at the ventricles, carried by the Purkinje fibers, the ventricles contract and send blood to, well, it depends which side you're talking about. Okay, to a pulmonary artery on the right side and to aorta on the left side. Okay, and of course, doing that, we have to make sure that the semilunar valves open. All right, so basically it's like the backwards of what's happening in the first phase. This looks a little bit messy. It's probably a good idea if you, reorganize it after class and write it in a way that makes more sense to you. But these are the key things that are happening, as the signal is spreading to different parts of the heart. Anybody have any question? Excuse me. Okay, so that is that. All right, I will move back to our lecture now. All those notes will be posted, right, after class today. Hold on. Slideshow. It's showing up on my other screen. Give me a second. Let me see what's going on here. Primary monitor. Okay, there we go. So this basically describe the whole process for you if you need to read it on your own. We already explained it just now, so I'm not gonna go through it again. I know it says like this is the AV bundle, and then in the label we also call this the AV bundle. Don't worry too much about the AV bundle. The key things for us to remember is SA no, AV no, and then Purkinje. Okay, that's the flow of information that you should know. Okay, everything else in between they call it AV bundles. So an artificial pacemaker is needed when the conduction system fails. Okay, if your heart is not able to set its own pace, then it could lead to arrhythmia. Arrhythmia is basically irregular heartbeat. Okay, so it might go really fast, boom, 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 or it will go really, really slow, depending on the type of arrhythmia you have. And sometimes is irregular in the sense that there is some delay in the signal conduction system. And you know, depending on the severity of the arrhythmia, you might need to either take medication or implant a pacemaker. So the pacemaker basically is a electrical device that regularly sends out electrical signal that would override your own faulty pacemaker, and therefore you rely on it to set your heart rate for you. Now, to detect the impulses that are generated by your heart, we can use a non-invasive method called ECG. Okay, non-invasive means they don't have to stick anything inside your body, no needles, no, you know, don't have to cut you open, basically. Okay, so that is called an ECG, an electrocardiograms. It's something that's done, you know, quite commonly to a lot of people. You might even had an ECG done on you before, and they basically stick these little stickers. They are like electroplates that measures electrical signal, and on the screen you would see a pattern like this. Okay, even if you have not had an ECG done on you before, you've probably seen something like that on TV, right? Like someone is at the hospital and then next to them there's a monitor that goes beep beep, right? That creates this wave. That's a ECG signal. Okay, so there are different parts to these waves, and they correspond to different phases of the cardiac cycle. And you know, if the readout shows something that is different from the normal one, then the doctors can make diagnosis, okay, and pick up things such as arrhythmia. So let's see which part of the curve corresponds to what phase of the cardiac cycle. When the SA note, your natural pacemaker, creates that initial signal, it causes a small bump. Okay, so this small bump at the front, right, that corresponds to atria contraction. Okay, and then there is a little bit of a delay here. Okay, that delay, okay, that's this flat line here, that's waiting for the signal. The impulse signal is spreading down the septum. Okay, so it takes some time to travel down the septum. The septum don't contract, right? It's the ventricles that contract. And then when it finally goes up through the Purkinje fibers, it will cause your ventricle to create a forceful squeeze, and that's what this peak is representing. Okay, so this is ventricles contraction. Okay, and then don't worry about the rest. The rest is just the heart resetting itself. We're not going to worry about it. Okay, so contraction of atria at the beginning, a little bit of a lag time as the signal is going down to the ventricles, and finally you have that big spike, which correspond to ventricular contraction. Okay. So you don't have to be a doctor to know what's wrong with these ECG. Okay, so the first one, right? What does it look like? What's the problem here? If you look at this one compared to like a normal one. I guess is I should show you like a normal reading, right? With many cycles. But you know, just any guess here? If you take a look at this, what do you think the problem is here?
There's not a lot of firing going on.
Yeah, exactly, right. Thank you, Leah. There's not a lot of you know, signals being sent out, right? And the heart rate is actually really, really slow. So that's bradycardia. You don't have to know these names, okay? I'm just writing it here to show you. So bradycardia. Brady means really slow. So in this case, really slow heart rate, right? 46 beats per minute. I mean, that might be a good thing, might be a bad thing, depending if the person is subdued with you know, anesthesia, for example, maybe it's okay for the heart rate to slow down a little bit. But for a normal person, 46 beats per minute, that is definitely way too low. Okay. This one is way too many. So that's tachycardia. Okay. It's too fast, firing too fast. 136 beats per minute, definitely not normal. And this one is just terrible. Okay, this is ventricular fibrillation. This is when the ventricles doesn't know what's going on. It just keeps on randomly squeezing and contracting with no rhythm at all. If this is not dealt with immediately, the person could go into cardiac arrest you know, could potentially die. So in this situation, that's when you need the defibrillator. Okay, the defibrillator will give a shock to your system, to the heart electrical system, and that shock sometimes will reset your SA node, and then it will be okay again. You know, it's the thing that you see on TV, right? Where the person the doctor rubbed the pads together and say clear, and then they shock the person in the chest, right? But nowadays, like even if you ever on campus, right, or at the mall, or you know, pretty much everywhere, they have these AED, right? So you don't have to know how to use them per se. Okay, you just literally stick the sticker on, press a button, and then the machine does the rest, right? And that is for emergency use to help people who might be undergoing ventricular fibrillation to reset their heart. Okay. All right, let's do some polls and see how you guys are doing. Go ahead. Okay, thank you for participating. We learned about cardiac output last class. Cardiac output is heart rate multiplied by stroke volume. Okay, heart rate is beats per minute. Stroke volume is how much blood you pump out per beat. Okay, volume as in like milliliters. If you cross them out and multiply it, then the unit for cardiac output is milliliter per minute. And so the correct answer is A. All right, so good job, the three of you who got that. And for the other people, that's okay. Hopefully I've convinced you what the unit for cardiac output is. Okay, so cardiac output is a measurement that kind of give you an idea of how healthy your heart is, right? Okay, this one, we talked about that last time. Let's see if you remember it. Okay, yeah, I think all of you got it. That's right. The when you're exercising, your heart rate goes up, and therefore your cardiac output would increase. Okay, let's move on to blood vessels. Function of blood vessels. There are three types of blood vessels, as you probably know by now. There are arteries that takes blood away from the heart. There are capillaries where gas exchange occurs, amongst other things, okay? And then there are the veins that return them back to the heart. And of course, right, in between the arteries and the capillaries, there are the smaller versions of arteries. We call them arterioles. And then between the capillaries and veins, we have the venules, right? And these are like smaller versions of the big vessels. Function of blood vessels. Okay, some of these are pretty self-explanatory. They transport blood and its content, right? So inside blood, there are a lot of stuff, right? Actually, we'll learn about blood next week, but just for the sake of talking, what are some of the stuff in blood anyways? What is in the blood? I mean, there's water, there's blood cells, but what other things are in blood? Anybody want to give me some ideas? You can type it out. You can shout it out. Sorry, what is it?
Nutrients.
Yeah, there are nutrients, right? Things like glucose. There are some amino acids, right? What else are there? Oxygen. Yeah, Beatrice said oxygen. That's correct. Carbon dioxide, right? So not just nutrients, right? There are waste product as well, right? So the blood kind of take these things to all over the body and deposit the nutrients in the body where it's needed, and then the waste would be excreted. Maybe through the lungs, you breathe it out. Some of it will be filtered at the liver. Some of it will be filtered at the kidneys, for example. So, blood, there's many things in it, and all these components have vital function in the body, and the vessel just kind of takes them all over the place. Okay, so at the capillaries, there is going to be exchange of gases in pulmonary capillaries and exchange of gases and nutrients at systemic capillaries. So I do want to mention the difference between pulmonary capillaries and systemic capillaries. Pulmonary means at the lungs, right? So at the lungs, right? We briefly mentioned last time that the O2 would enter the blood, enters blood, and CO2 leaves blood for the pulmonary capillaries. Okay, because you want to breathe out carbon dioxide, right, out the lungs. But at systemic capillaries, at the body, say, in the digestive tract, for example, when the blood goes to the digestive tract, the O2 would now leave the blood so that your digestive system can use it. And then it will pick up carbon dioxide from your digestive system, and it will enter the blood. So the direction of gas exchange is dependent on which part of your body you're talking about. Right? It's not always oxygen coming in or leaving. It depends on whether you're, at the lungs or whether you are at like an organ system. Okay. Another function is the ability to regulate blood pressure. We'll talk more about this in a bit, but the blood vessels can change size. Okay, so if you look at a blood vessel like this tissue paper roll here, okay, like the inside is called the lumen, L-U-M-E-N. The lumen is the inside of the blood vessel. If it's big, then the pressure would be lower, but if it squeezes and becomes smaller, then the blood pressure will go up. So depending on your body's blood pressure, the arteries and can change the diameter to either increase the blood pressure or lower the blood pressure. Do you guys have an example of where or why your blood pressure might drop? Doesn't have to be like serious health problem, like everyday life things that we do sometimes cause our blood pressure to drop. Any example?
Shock?
You can go in shock and then your blood pressure can drop. Anything else? Something that's more common occurrences? If you're relaxed, yeah, okay. Sometimes.
If you're dehydrated?
Yes, when you're dehydrated, okay? When you don't drink enough water, then your blood volume goes down, and then your blood pressure actually goes down as well, right? So in those cases, your blood vessels will actually constrict a little bit to help bring the blood pressure back up, right? Another one is like blood loss, right? If someone has an accident and they lose a lot of blood, you know, then their blood pressure could go down. But to temporarily bring it back up, your blood vessel can squeeze. That's the point I was trying to make. You can also, the blood vessels can also direct blood flow to parts of the body that that is needing the blood the most. Okay, so like let's say you had a big meal and then you're just sitting at the couch watching TV, then the blood vessels will divert the blood to the digestive system, right, to help digest your meal. At that time, because you're just sitting on the couch, you don't really need a lot of blood going to your muscles. Okay, so they will restrict the blood flow to the muscles and you know, divert it mainly to a digestive system. If you are you know, running around, jogging, for example, then at that instance, you need more blood to your muscles, your leg muscles, and you might want to divert more blood to your lungs so that you can pick up more oxygen. At those instances, you would decrease the blood flow to your digestive system, because when you're jogging, you're not really going to digest your food, right? So depending on the body's need, the blood vessels can divert the blood accordingly. So how does it divert the blood? Well, to keep things very simple, here is an artery, okay, and here is a vein, and this is a capillary network in between. All right. So normally, under normal circumstances, like no restriction, then the blood would come in, and then it would just, you know, infuse the entire network, right? Can you visualize that? Okay. Coming like that. So the entire network is 100% filled with blood. But let's say, you know, whatever this part of the body is, let's say, you know, it's the muscle, and we're just sitting on the couch watching TV. We don't need as much blood going through it. And so what happens is there are these little valves or sphincters. Sphincter is just another word for valves. Then, you know, they might close them off. They might close it off. So let me just choose another color here. Okay, so you can close off this sphincter, close off this sphincter, close it off. And then what happens is now the blood, instead of, you know, filling up the entire network like before, it will just go right past it. Okay, like that. So not as much blood going through it, right? But you don't cut it off completely. I mean, cut it off completely, then the muscle dies, right? Or whatever the part of the organ this is at will die off, right? You cannot 100% cut off, but you can reduce the flow. Okay, and instead of filling up the entire bed, right, you just let it pass through. So you get some nutrients, you get some oxygen, but not at full capacity. Does that make sense? I hope it does. So over here is just a picture of arteries and veins, and you can see that they are quite different. Okay, now first of all, they are not literally blue and red. Okay, this is a picture that has been stained to contrast, highlight the differences. So can you tell me some of the differences that you can observe based on this picture? Difference between the arteries and the vein?
Thickness?
Yep, definitely. The artery is thicker, right? So artery is thicker.
The size.
The size, that's right. So not only is it thicker, but it also has a smaller lumen, right? Lumen is the inside, huh? Smaller lumen. Right? So generally speaking, arteries, they are thicker but smaller. But for veins, they have thinner walls and they're bigger. Okay. And I will explain why that is the case later on. Okay, moving on. All right, we're going to talk about capillary exchange, which is a really big concept, okay, that I'm going to explain. So please, I'm going to give you a moment to get some paper, and you probably need like 3 different colors, okay? Just give you like a minute to get those ready. We're going to take a really big note. All right. All right, hopefully you're ready by now. So this is a note called capillary exchange. what is capillary exchange? Well, basically, capillary exchange talks about the exchange of substances across the capillary wall. All right. In plain English, is basically what goes in and what comes out of the capillaries. Okay. So we're going to look at three factors that affect capillary exchange. Okay, I'm going to see three things that will influence what goes in and what comes out of the capillaries. Now, the first one is quite straightforward. Okay, you learned this last semester. Diffusion. Diffusion. And hopefully we all know what diffusion means. It is the spontaneous movement of substance from high concentration. That's what the square bracket means. Okay, square bracket means concentration. High concentration to low concentration. Things naturally move from an area that is high concentration to an area of low concentration. Okay, you open a bottle of perfume at the back of the classroom. Eventually, the whole classroom will be able to smell it because it's naturally moving from high concentration, the bottle, to low concentration, the hemisphere. Okay, so let's illustrate this, with a picture. So let's say we have a blood vessel right here. There's a blood vessel, and this is a capillary, okay? Capillary. On one side, it will be connected to the arterial. So this is arterial side. And then on the other side, it's going to be connected to a venule. We would call this the venous side. Okay. Just going to give you a moment to catch up. Again, if you could, just give me like a thumbs up when you're ready to move on. Okay. Sometimes I know it's hard to follow me if I'm going too fast. Do you guys not know where the thumbs up is? I mean, some of you are doing it, but just wondering if everybody knows where that thumbs up button is. All right. So, I'm going to use these, thank you. I'm going to use these red balls, dots, to represent oxygen. Okay, so maybe you have a lot of oxygen here in the capillary. Okay. And then maybe you have a little bit of carbon dioxide. Okay, so these are like CO2, and then these are like O2. Right, and then on in the surrounding area, this would be like the tissue. Okay, when I say tissue, I'm talking about like an organ, right? Maybe we're talking about the kidney tissue. Maybe we're talking about stomach tissue. Whatever, it doesn't really matter. Okay, so it's just the surrounding area. Okay, and so maybe, just maybe, there will be less oxygen, right? Because it's being used up. And you learned about cellular respiration last semester. So, your tissue, your cell's going to take the oxygen, reacting it with glucose, and then ending up producing carbon dioxide, right? So you might have a lot of carbon dioxide building up here. Okay, so as the blood passes through the capillary, just by diffusion alone, okay, you don't have to do anything. It doesn't require ATP or anything like that. Just by diffusion alone, the oxygen will leave the capillary like that, right? From high concentration to low concentration, right? And similarly, the carbon dioxide will come in. Okay, so these things are moving down its concentration gradient, just spontaneously. Okay, so afterwards, the blood will become flipped, right, so to speak. It's going to be different than before because of all the oxygen leaving. Now you will have less oxygen, but then you will have more carbon dioxide, right? Does that make sense? Right, so you have high, I should probably put it in here. High O2 and then low O2. And afterwards, you would have low O2 because they all left. And then the outside would be like high O2, right? And then there would be the opposite for carbon dioxide. You would have like low carbon dioxide. And then this would be high carbon dioxide. And afterwards, it will be high carbon dioxide. And then outside would be low CO2, right? So this is an example of systemic circulation, right? Remember, there are two circuits, right? There's the pulmonary circuit, and then there's the systemic circuit. This would be a systemic circuit where the blood went from red to blue, right? Because it picks up all the carbon dioxide and deposit all the oxygen. Please ask any question if you need clarification before I move on to the next two, because the next two is the confusing one. This is the easy one, but I just want to make sure we're all okay before moving on.
Wouldn't this be not systematic but pulmonary because the high O2 is within the capillary and it leaves?
Okay, yeah, that's a good question. Okay, so look, let's think about over here. Okay, is the blood in the capillary? Would you say it's like red blood or blue blood in this on this side here in the arterial side?
Like red?
Yeah, because of high oxygen content. Do you agree? So where does this high oxygen content blood came from? Well, probably it came from the aorta, right? It wouldn't have came from the lungs, because if it came from the lung, sorry, it wouldn't. We're not going to be going to the lungs, because the only time we send blood to the lungs is when we don't have enough oxygen in the blood, right? So we can reoxygenate it, right? So if you agree that this is high oxygen blood, then we must be sending this blood to the body so the body can use it, right? So when I say tissue here, I'm talking about like, like I said, like a stomach, for example. So the stomach needs oxygen. That's why the oxygen will be coming out of the blood and into the tissue. Does that answer your question?
Yes, it does.
Okay,
Thank
great.
you.
Someone send me a private message. If low O2 will be going outside, and then high O2 will be inside. Okay, so over here, right, because we have high oxygen on the inside and low oxygen on the outside, that's why the oxygen will be leaving, right? Okay. After the oxygen leaves, then that's why we have low oxygen now. Okay, they all left. Okay, so now the surrounding area will have high O2, and then these oxygen will now be used up by the cells, right? And so, it will basically go back to this situation once the oxygen is being used up. Okay. All right. So that is the first factor. Now, just in case you forgot why we are talking about this, we're talking about capillary exchange. We're talking about factors that would determine what goes in and what comes out. Okay. So the first factor is diffusion. Right. That's really simple. Depending on which side has more of what, then that thing would either go out or come in. All right. Let's take a look at the next two factors. All right. So the second factor is blood pressure. Factor number 2 is blood pressure. Okay. What exactly is blood pressure anyway? Well, blood pressure is the force of blood pushing against vessel wall. Okay. If you think about a balloon, right? You blow up a balloon, tie it up, and you touch the balloon, it feels like there's pressure inside, right? Well, what's creating that pressure? Well, that's basically like air molecules pushing on the walls of the balloon. Right? So it's no different when we talk about blood. Okay? So you have your blood vessel here. Right? Blood pressure is essentially the blood pushing on the vessel wall. Okay, and generally speaking, the more blood you have, as in like the higher the volume, the higher the force of pushing will be, and you would have a higher blood pressure. Okay, so blood pressure is the force of blood pushing against vessel wall. Okay, so what's creating the blood pressure? Well, I want you to imagine this, okay, and maybe you have had experience with this before. Let's think of like do you guys know what I'm drawing? This is a faucet, right? Like a water tap, okay? And I'm gonna hook it up to a really long hose, okay? And then I'm gonna poke holes in the hose. All right. I will turn this on to maximum. Turn it all the way. Now, someone tell me what will happen. It sounds like a rhetorical question, but not really. Okay, I just want to see if you guys understand, if you can visualize this. What will happen if I turn on the tap all the way? Well, hopefully you're thinking, well, of course, water is going to come out, right? Okay, so water will go like that, come out, right? But do you expect it to be the same amount of water coming out as we move along the hose? Like, what if we are like all the way at the very end? Do you expect it to be as strong as the beginning?
No.
No, right? Okay, so like as you move further and further away from the source, the water will still be coming out. Okay, but maybe with less intensity, right? By the time you get to the end, maybe water is just kind of sipping out. Okay, so I want you to understand this and be able to visualize it. Okay. Now, we're going to replace the faucet with the heart. Okay. And then we're going to have vessels coming out of the heart. Okay. So this is the aorta. Okay, and then maybe you have some smaller artery, arterial, capillaries, venules, and veins, right? Right. So what about the blood pressure? Well, at the beginning, you will have very high blood pressure, right? Pushing things. Okay, but as you go further and further along, right, that blood pressure will drop significantly. Does that make sense? Okay, so really high blood pressure. And then really low blood pressure for the same reason as the water hose. Okay, the further away you are from the source that's creating the pressure, the lower the pressure will be. All right. Let's draw another capillary. Okay, so there we go. Make sure this one is big, okay? We are gonna draw a lot of stuff on it. Draw over here. Okay. No, hold on, okay? Don't just copy it just yet. Okay, give me 1 second. If you're using pencil, that would be good. But if you're using pen, just pause for a second, okay? Watch. This is capillary, okay? This is arterial side, and then this is venous side. Okay. Now I didn't really tell you about this earlier, but capillaries the walls, they are not like sealed, okay, 100% solid like the tissue paper core here. They are actually porous, okay? So they actually let me see where is my right there. Okay, they actually are leaky. Okay, so there are gaps in it. So when you draw it, make sure you draw like dotted lines and not like solid lines. Okay, so capillaries, they are leaky. Okay, leaky walls. They just are. Okay, you just have to believe me. I'm not gonna explain too much about it. Okay. So, what is the effect of blood pressure on capillary exchange? Well, as I try to explain to you here, the blood pressure is going to be quite high on the arterial side, and it will be pushing things out. It'll be pushing things out like that. Okay. But as you move towards the venous side, right, the blood vessel is going to, the blood pressure rather is going to drop. Okay, so on the arterial side, maybe the blood pressure, these are just hypothetical numbers that helps you understand what's going on, okay? So the blood pressure over here might be 30 millimeter mercury, mmHg. That's the unit for measuring blood pressure. But on the venous side, the blood pressure might be, let's say, 15 mmHg, okay? But regardless of the number, right? The effect of blood pressure on capillary exchange is that, I'll write it down here, blood pressure pushes fluid out of capillary. Okay, so push things out. Not blood. We're not going to be leaking blood out of a capillary. That's like hemorrhage. We don't want that. We are just pushing the fluid component of the blood out of the capillaries. Okay, so it's mostly water. There might be some dissolved nutrients in it. Okay, and these things will be oozing out from your capillary. Any question? All right, so that's the second factor. Okay. Next, we can talk about the third factor that affects capillary exchange. Keep this diagram handy. We're going to add more things to it in just a second. The third one is called osmotic. Osmotic pressure. What is osmotic pressure? All right. Let's quickly think about this one. I mean, if you were with me last semester, I spent quite a bit of time trying to convince you about this scenario. But you know what? Let's just refresh our memory on that. Let's say I have two chambers here, one on the left, one on the right, and then I have some solutes in it. Okay, some solutes. You know what? That's not big enough. Let me just make it bigger right there. Okay, we have some solutes here. Okay, and then we also have some solutes here. And then let's say the chambers, they are filled with water. Okay, filled with water. Okay, let's make this you know what? I'm just gonna tell you it's filled with water. You don't have to draw the water. And here is a membrane. Here is a selectively permeable membrane, which only allows water to pass through. Okay. So obviously you have a high concentration here on the left with all these purple solutes, and then you have a low concentration here. So my question to you is, let's call this A, let's call this B. My question to you is, will the water move from chamber A to chamber B, chamber B to chamber A? Right? What is going to happen? Please let me know in the poll. And my question is, if I leave this alone, which way will water move? From A to B or B to A? All right, last call. Anyone else want to log in their answer before I reveal? Okay. So, we have quite a bit of people choose A and then quite a bit of people choose B. So which is it? Well, the key thing to know here is that the membrane will only allow water to pass through. Okay, these purple balls can't go anywhere. Okay, so water will actually go from the right side to the left side. So that is the correct answer. Okay, water moves towards the area of higher concentration through osmosis. Okay, the best way to visualize this is to imagine, or like you can actually try it out if you want. take a spoon of salt and then put it in your mouth and hold it there. Okay, don't swallow, just put it in your mouth, and then wait to see what happens. Well, you probably can imagine what happens, right? If you have the spoon of salt in your mouth, it's gonna suck the saliva out of your mouth, right? And then your mouth becomes like watery because of the salt, right? So the salt is actually drawing water from the surrounding area. In the same way, because we have more solute here, more salt, right? It's going to draw the water towards that direction. Okay, and this is what osmotic pressure is. Osmotic pressure is created as a result of a difference in solute concentration. So this is created by difference in solute concentration across the vessel wall. Okay, so you might have more solute on the inside than outside, and then that will draw fluid towards the inside. So for those of you who chose A, have I convinced you that the answer is B? Anybody want to ask any questions about this so I can clarify for you? Okay. So osmotic pressure, right, created by difference in solute concentration across the vessel wall. So what is the effect of osmotic pressure? Well, earlier I kind of asked you guys, right, like what is in the in blood, and you guys gave me a lot of excellent answers, right? So there might be like some nutrients, there might be some waste products, some cells, right, some proteins. And so the blood is packed with stuff, and all these stuff that we mentioned, they contribute to the concentration of the blood. Okay, so on the inside, all these dots, right? These are like proteins, blood cells, right? Electrolytes, perhaps. All these things contribute to your blood concentration. And generally speaking, it is higher on the inside compared to the surrounding tissue. Okay, so you have more stuff in the blood than on the outside. And so based on this, are we going to be drawing fluid into the capillary, or are we going to be pushing fluid out of the capillary? Let's do another poll here just to see if you're with me. Okay, my question is, are we going, because of this difference here, are we going to be A, drawing fluid into the capillary, or B, out of the capillary? Which way is it going to go? In or out? Please let me know your choice. Oh dear. We are still at 50-50 here. Okay. Let me try to explain this again. I think I understand why you guys think it's going to be going out. You are focusing on the green dots, okay? The people who chose B, I think you're focusing on the green dots. You're like, oh, of course the green's going to go out because it goes from high concentration to low concentration, right? We're not talking about the green dots, okay? Because, you know, these proteins, these blood cells, they are too big to leave the capillaries. So we're just focusing on the fluid, okay? The fluid, which is mostly water, right? Will the fluid go into the capillaries or out of the capillary? Think of the spoon of salt that I talk about, right? So like you have a lot of salt here compared to the outside. So that's actually going to be drawing fluid into your blood vessel. Do you understand that? It's absolutely crucial that you understand that. A lot of the things that we talk about later on will depend on this concept. Okay, so I'm just going to pause again and then see if you have any question. Those of you who chose B particularly, have I convinced you that it should be A, that fluid is going to be coming out? Okay, so let me write this down for you. I'm sorry it's getting really crowded here, but I guess I'll just put it over here. OP, and obviously you got to reorganize your notes after class, right? You maybe watch it again and then, you know, redraw it and whatnot, okay? So OP, osmotic pressure, draws fluid into capillary. Okay? Unlike blood pressure, which drops as we go from one side to the other, the concentration of the blood does not vary substantially from one side to the other. And so the osmotic pressure created by that difference is actually going to be the same as we move from one side to the other. Okay, I try to make them the same length, okay? It's very difficult to draw on the computer, but I hopefully you get the idea. Okay, so OP here, osmotic pressure on the arterial side, about 20 mmHg. Again, these are just hypothetical numbers. And on the right side, right, it's the same number. I told you there is not a significant change in the salt concentration in blood concentration from one side to the other. That's why it's the same on both sides. Okay. So I'm gonna pause here for a second. I'm gonna let you just go on a break and maybe put some salt in your mouth to help understand this concept. And then when we come back, I will finish explaining what's going on here. Okay, so 5-minute break. We'll resume at 9:45. Okay, see you in a bit. Hello, it's me again. Let's keep going. So let's summarize everything we talked about so far. Let's go back to our study guide, and there's a chart right here. Page 2, I think. Yeah, right there. Page 3, maybe, perhaps. Question number 11. Let's just put everything together in a nice chart so we have an easy way to refer to it if we need to. So on the left side right here, we have osmotic pressure. Let's just stick with the same color code as we had before. So osmotic pressure It is created by concentration. Concentration difference across capillary wall, vessel wall. Okay, and I'm just going to put a side note here. Fluid is drawn towards higher concentration. Okay. That's the osmotic pressure. For blood pressure, it is created by basically contraction of the heart, right? Contraction of heart. And as a result of that contraction, you're going to have fluid pushing against vessel wall. I know you wrote all these down already when we took the notes, but it's just nicer to have it all in the table, right? Effects on capillary exchange. Is it drawing fluid into the capillary or pushing it out? And just pause for a second and think about that. You don't have to answer me. Just answer to yourself, okay? Osmotic pressure, is it pushing it out or drawing it in? Hopefully, if you think back to the diagram we drew, you would know that the effect of osmotic pressure on capillary exchange is that it will draw fluid into the capillary. When I say fluid, I'm talking about mostly water, but with stuff that are dissolved in it as well. And on the other hand, the red arrows, right? Like the water holes that I drew for you, the blood pressure will push fluid out of the capillary. Capillary. Okay. Let's take a look at the arterial side versus the venous side. For osmotic pressure, which side is higher? Is it the arterial side or is it the venous side? What do you think for the osmotic pressure? The green arrows, which side is higher?
Same.
Yes, you didn't get tricked. Very good. Okay, it's the same on both sides. Okay, so OP is the same on both sides. Whereas for the blood pressure, BP is higher on arterial side. Okay, that's that. All right. If you haven't finished copying, that's okay. I'll post this after class. We do have to move on. Now, just one last thing I want to talk about here before we finish with the capillary exchange. Let's take a look at this diagram again, the one that we drew. And I want to focus on the numbers here. So by now, I hope you understand one is pushing things out, one is drawing the fluid back in, and it's kind of like this opposing forces, right? These two opposing forces. The BP is pushing things out, the OP is trying to bring the fluid in, and depending which one is stronger, the overall net effect will be different. So on the arterial side, we have 30 mmHg pushing things out and 20 mmHg bringing it in. And so if you do the calculation over here, the net pressure is actually going to be 10 mmHg in what direction? Again, just think about it to yourself. Is the net pressure going to be ending up pushing things out or bringing things in? Well, the red arrow is longer, right? So 10 mmHg pushing things out. So on the arterial side, you would have fluid loss. Okay, so you will have fluid coming out into the tissue. On the other side, the osmotic pressure, because it did not change, it stayed at 20, whereas the BP actually dropped to 15. And so if you do the calculation here, the net pressure is going to be 5 mmHg, but it's going to be inside now, right? Because the green one is longer, and so you will have fluid gain. Okay, so now you're going to be bringing some of it back into the blood vessel. Do you understand how you will be losing fluid and then you will be gaining some of it back? Now I keep on saying some of it, right? So the pressure here is 10, the difference is 10, and the difference here is only 5. So you're not going to be able to bring in everything, okay? You will end up losing more than what you were able to bring back in. Do you understand that? Okay, you might wonder, well, what happened to all that leftover fluid? Where do they go? Well, that's something we'll talk about in lecture 2, all right? That actually is an important part of your immune system. Okay, the ability to not bring everything back in. Okay, your body deliberately lose more than you bring in, and something's going to happen to those excess fluid, and we'll talk about all that in lecture 2. But yeah, that's the end of the story. Okay, one last time, any questions before we move on to the next big story? No, you all with me? Okay. Let's go back to our PowerPoint then. Okay, let's do some quick poll questions. Now try to answer these without looking at your notes, okay? We literally just roll it down. So let's see if you can do it from memory. Go ahead. Okay, I have 10 answers. Can I get a little bit more before I close the poll? We have 21 people here, so. All right. Yeah, most of you got it right. The BP is greater than OP on the arterial side, and so you are going to be ending up pushing things out. Let's do another one here. Right. Again, most of you got it right. OP is the green arrows. The green arrows don't change from one side to the other. And so the correct answer is C. All right, moving on. Next concept is venous return. Right? Venous return is basically taking blood and returning it to the heart. That's what it means. Okay. Now it's not as simple as you might thought, because there are two major challenges that we have to overcome when it comes to venous return. So this is the person and this is the heart. And you know, for blood that are below the heart, they have to actually go up, right, to get back to the heart. And so, you know, you have to overcome gravity, right? That's one big challenge. And number two, you know, we are in the veins now. And if you think about the water hose, that picture that I drew for you, when you're so far away from the heart, when you're in the veins, the pressure is really, really low. So low pressure in veins. Okay. And when things are low pressure, when fluid meaning like liquid or gas, when they're in a low pressure system, they don't tend to move very fast. Okay. And so, you know, speed and pressure is a problem, and to make it worse, we have to go upwards against gravity. So how do we overcome these problems in venous return? We're going to look at two ways. Okay, number one, this would be like a vein in the leg, for example. And so every time you walk around, move your leg muscle, your muscle actually serves as a bit of a pump for the blood. So when you contract, when you squeeze those calf muscles, it will cause the blood to get squirted upwards towards the heart. But you know, our legs don't stay contracted continuously, right? Like, think about walking. Okay, as long as you lift you up, then your calf muscle would relax. So as soon as the muscle relax, then the blood will just fall back down. Okay, but they don't fall all the way back down to your foot because of the presence of valves. So here you see in the veins there are these valves. So the valves will open when your calf muscle contracts to allow the blood to go up. But as soon as the calf muscle relaxes, then the valve will close and the blood will come down and the valves will be able to catch it. So next time when you contract again, it will squirt the blood upward again, and then it will just climb a little bit and a little bit and a little bit at a time, and until it gets back to the heart. Okay, so to compensate for the low pressure, the muscle will create that extra squeeze, and to compensate for going against gravity, we have these valves that will catch the blood from falling, so they don't go all the way back down to your foot or something, right? You just go a little bit and a little bit at a time, and eventually you will make it back to the heart. Now, if you ever been on a long flight, okay, and you know, you decided to take your shoes off and then make yourself comfortable, have you ever not be able to put your feet back into the shoe afterwards? Like I mean, okay, that might be a little bit extreme, but like when you try to put your shoes back on at the end of the flight, it feels like it's tighter fit, right? Like you have a harder time putting on your shoes, right? So you might have had that experience, or you might have heard of people who have had that experience, right? That's because when you're not moving your legs, when you're just sitting in the plane for like 10-12 hours, this mechanism is not really working. So the blood is not really returning to the heart as much as it should have been. And so if you think about the capillary exchange thing that we were talking about earlier, right? And this would be like in the foot, for example, because of the blood pooling in the foot area, all those extra blood is now increasing your blood pressure, right? So now you're pushing even more fluid out of your capillary than you otherwise would. Your osmotic pressure does not change, okay? The blood concentration does not change. But because of the pooling of the blood, because you're not moving around, you're not using this method to return all the blood to the heart, what happens is all those extra fluid would increase your blood pressure at the capillaries in the foot, and all these extra fluid is now accumulating in the foot, and the foot actually swells up a little bit, okay? And so you might have a hard time putting your shoes back on. It doesn't have to be an airplane. If you're a mother, you might remember during pregnancy, your foot might actually got a little bit bigger than pre-pregnancy. Your shoes might be a little bit too snug, and it's the same reason. Okay, so if you think about the mother, right, and now the mother is pregnant with the baby, so all that extra pressure is causing difficulty for the blood to return to the heart, and the same mechanism is occurring, causing the foot to swell up a little bit. Okay, so that is a venous return. Now sometimes you might have these, or you might know people who have these. These are called varicose vein, and that's when the valves are damaged. Okay, the valves and the veins are damaged, and the blood is not able to empty completely and return to the heart, and they start pooling in the legs, and you can see all these veins kind of bulging out. And again, like this could happen during pregnancy as well, right? Might not be full-fledged varicose vein. It could just be like those spider veins, and sometimes the doctor will tell you to put on those elastic stocking, right? So that's elasticity, that extra pressure from the elastic stocking would actually help return some of the blood back to the heart. Okay, so there you have it, varicose vein. Okay, let's talk more about blood pressure. We already established that blood pressure is created by the blood pushing against the vessel wall, and the source of the blood pressure is actually the ventricular contraction. We learned about systole, which is contraction, and diastole, which is relaxation. So depending whether you measure the blood pressure during systole or diastole, the numbers are going to be different. So systolic pressure is usually the higher one, and in many textbooks, even if you go to like Shoppers Drug Mart, like the chart would tell you, 120 mmHg is like the normal, and then the diastolic pressure is 80 mmHg. But it's important to not read too much into these numbers. Okay, these are just like general guidelines. Okay, what's considered as healthy or not depends on your age, for example. Okay, it depends on your physical condition, and it depends on your own history, right? Okay, some people just naturally have lower blood pressure, and that's okay, and some people have slightly elevated blood pressure, and that might be okay as well. So these 120, 80, it's not set in stone. It's not like if you have 125, then you have high blood pressure necessarily. Okay, it really depends on the context and the background of the person. So blood pressure depends on many factors, and here are some common ones that we can talk about. Total blood volume. So in general, the more blood you have, higher blood volume means higher blood pressure. Okay, higher blood volume means higher blood pressure. The more blood you have, the higher blood pressure there will be. So like if you have dehydration, we talk about, that causes your blood volume to go down, and therefore your blood pressure will drop. Okay, severe dehydration is dangerous because your blood pressure can drop so much that you might not have enough circulation, and that could be lethal, of course. Some people who have chronic high blood pressure, okay, if they actually I mean, your body has mechanisms that will bring the blood pressure back down if it's too high. We learned about that in lecture 9. Okay, but if those mechanisms fail, then the person will have chronic. Chronic means like ongoing. They will have ongoing high blood pressure. Does anybody know anyone who has high blood pressure and then know what kind of drugs that they have to take? Like, maybe I'll just tell you what it is and ask you if anybody heard about it. Anybody heard about like water pills? by any chance? Water pills. People with high blood pressure, sometimes they have to take water pills. Anybody know what that might be? Or even heard about it?
Aren't they diuretics?
Yes. What is diuretic? Can you explain to me?
Makes you pee.
Yes, that's exactly what it is. Okay, very good. So a diuretic is a pill that makes you pee. Now, why would peeing help with the situation? Well, what is pee? Pee is basically water, right? Okay, so if you pee more, your blood volume would go down accordingly. Okay, so for people who suffer from high blood pressure, peeing more will actually lower their blood volume and therefore lower their blood pressure. Okay, so that's one way of combating high blood pressure.
Professor, doesn't it also have to do with the sodium in the pee?
Uh,
Or not
yes,
really?
well, the diuretic doesn't really change the sodium. It causes you to pee more through other means. But you're absolutely correct. Your body's own mechanism, again, we will learn in lecture 9. When you have high blood pressure, your body will actually deliberately put more salt into the pee, so that the salt in the pee will draw more water from your body, and therefore lower the blood pressure. Okay, so just hang tight until lecture 9. We're going to learn all the glory details by then. But in short, you are absolutely correct. Stroke volume. Stroke volume also affects blood pressure. If you have increase in stroke volume, then you will have increase in blood pressure. That perhaps is not too difficult to understand, right? Stroke volume is how much blood you squeeze out every time the heart beats, right? So if you squeeze out harder, you squeeze more blood out, you're going to have a higher blood pressure. Okay, that's self-explanatory. Vessel diameter. So is it a big vessel? Is it a small vessel? Generally speaking, if you increase diameter, right, so we call that vasodilation. Vasodilation. That means a bigger blood vessel, then you will have lower blood pressure. On the other hand, if you have vasoconstriction, if you're going to squeeze on your blood vessel, then you will have increased blood pressure. And of course, this is controlled automatically by your body. You have no conscious voluntary control over it, right? So your body will automatically adjust the vessel diameter as needed, so that you maintain a homeostasis for your blood pressure. Okay. I already told you blood pressure is measured in millimeter mercury, mmHg, and the normal is 120/80 mmHg. Okay. So now we're going to look at 3 graphs here, and hopefully it makes sense. First graph is the blood pressure, systemic blood pressure. So you measure it in the arm, right? Usually, okay. So it's possible to measure from the legs as well. But the blood pressure that is measured as it is going through your system. Okay, so you can see the top line here represent the systolic pressure, and then the bottom line here represent the diastolic pressure. And you can see when we are close to the heart, right, when we're looking at the aorta and the arteries, you can see the blood pressure actually fluctuates between the systolic and the diastolic, which makes sense. It means that the pressure is higher when the heart is contracting, and then the pressure drops when the heart is relaxing. Okay, and then it just keeps on fluctuating. But you can see the 2 lines actually merge and become one line when we get to the capillaries. Okay, what that means is at the capillaries, the blood pressure is the same, regardless of whether the heart is contracting or relaxing. Okay, you are so far away from the heart by now in the capillaries that you don't even feel the difference. Okay? Whether the heart contracts or relax, it no longer has an effect on the blood pressure at this point. It's just going to be a single number. All right. And earlier I was trying to tell you, right? Like, look at this capillary right here. Okay. At the beginning of the capillary, right? It's like about 30 mmHg, right? After you go through the capillary on the venous side, it is about like 15 mmHg, right? Do you remember the diagram? Right? When we drew the capillaries, I keep on saying, this is the arterial side, this is the venous side, and then, it's higher on one side and lower on the other side. Do you understand how it all connects? Right? So generally speaking, the blood pressure would continue to drop the further away you are from the heart. By the time you get to the vena cava, right, the superior vena cava, inferior vena cava, you virtually have no pressure, right? And then it would just like fall right into the right atrium. Okay.
Can you give me some examples of where, like, the locations of these capillaries?
Oh, capillaries are everywhere in the body. Every time you have a paper cut, you basically cut a capillary. Okay. No cell in the body is more than a few cells from a capillary. Okay. Because diffusion is not gonna be happening through a very long distance. So let's say I have a capillary here, right? Capillary network. Okay. I cannot have a cell like all the way out here. Okay, this will never get the oxygen. Like the oxygen comes out from the capillary, for example, and it will never make it to a cell that's so far away. So all the cells are going to be like, very close to a capillary network. If you have another cell all the way out here, then you will have another capillary network here to supply it for it. Okay. So basically your entire body is covered with these capillaries. And the next graph will really emphasize on this point. But for now, does that answer your question?
Yes.
Okay. So next graph. The two graphs here merge into one. So just kind of ignore this one for now. Okay, just focus on this one that's like fluctuating. So this is velocity versus blood flow. Okay, velocity is just fancy word for speed. Okay, speed. All right. So the speed, the velocity of blood flows, velocity. Sorry, I'm just gonna maybe write blood speed. If that makes sense. The speed of blood flow. Okay. Depends on two things. Okay. Number one, it depends on the blood pressure. And number two, it depends on diameter. Vessel diameter. Okay. So just try to use common sense. Okay. What's the relationship between blood pressure and speed? Do you think higher blood pressure is faster or higher blood pressure is slower? Right? What do you think? How does blood pressure affects the speed of blood flow? Faster or slower?
Higher blood pressure is faster.
Yeah, exactly, right? Higher blood pressure is faster. So increased BP equals faster. Okay. The next one, diameter. If I increase diameter, okay, now don't think about the pressure, just think about the diameter. If you increase diameter, is it going to be faster or slower? This one usually messes people up. Let's do a poll on it, okay? If you think it's faster, please choose A. If you think it's slower, please choose B. Big diameter, faster or slower? Okay. A lot of you say slower, but actually is faster. Okay, I know why you chose slower. I will explain in a second. Okay, let me rephrase. Give you another example. Okay, if you think about driving. Okay, are the cars able to go faster on a street with one lane or with like 4 lanes, 5 lanes. Okay, obviously, the more lanes, the faster the traffic will be, right? So same concept here. If the vessel is really big, blood is going to be able to go through faster than if it's really small. Do you understand that? Okay. Now I know why you chose B. You're like, but you said earlier that if the diameter is big, then the pressure will be low, and so it should be slower, right? Okay. Now we cannot look at so many things at a time. When we talk about relationship, we should just look at the single variable. So if we were talking about diameter, we completely ignore pressure. Okay. If we're talking about pressure, we ignore diameter. So if we just look at diameter alone without thinking about pressure, then high diameter would become faster, whereas low diameter would become slower. Okay. But in reality, it's a combination of both, right? Okay, so you can see here the velocity is actually going up and down, up and down, up and down at the beginning, and that totally makes sense because if you look back here, the pressure is going up and down at the beginning, right? And so we said here the speed is dependent on the blood pressure. So if blood pressure fluctuates, then speed is also going to fluctuate at the beginning. Okay, but then it will slow down substantially, and then it would be picking up again. So why is that the case? Well, in the artery, right? Arteries have smaller diameter. Remember that picture I showed you earlier with the red and the blue right next to each other, right? But the arteries have smaller diameter, and the veins have larger diameter, right? Okay. But the BP might have smaller diameter and higher BP because you're closer to the heart. Over here, you have lower BP, right? So that's why the speed is similar, a little bit higher, but like, you know, not too high. It's similar in the arteries compared to the veins. Even though the veins is larger, which would make the blood go faster, but then the pressure is lower, so that would slow it down. Initially, the arteries, they have really high blood pressure, so that would allow it to go faster, but because they're smaller, it will also cause it to go slower, right? So because of these opposing measurements, right, the speed in arteries and veins are similar, although a little bit higher in arteries. The takeaway point from this graph is that capillaries, that's where you have the slowest velocity. Okay, you need to slow down substantially at the capillaries in order for you to have enough time for gas exchange to occur. If you're just zooming by the capillaries, you won't have enough time for the oxygen to come in and, you know, for the carbon dioxide to move by diffusion. You must slow down, right, so that you could do that gas exchange. Any question about velocity? Okay, the last graph is total cross-sectional area. Now, what does that mean? It means if you lay out all the blood vessels of the body, which one would cover the most area? Okay, so it turns out capillaries will have the most total cross-sectional area. I was just telling you guys, right? Every inch of your body is covered by capillaries, okay? And they are everywhere. So even though individually the capillaries are really small, but because you have so much of them, you end up covering a lot of cross-sectional area. The aorta, right, you might remember I showed you a picture that I took, right, of the heart, and I said, you know, you stick your thumb in, you can still move around. So the aorta is really, really big, but you only have one of them. Okay, so the aorta doesn't actually cover too much cross-sectional area. Okay, so capillaries, even though they're small, but because you have a lot of them, it covers the most cross-sectional area. Okay, so those are the 3 graphs that relates to, you know, different types of blood vessels. Okay, so we talked about hypertension already. like I said, hypertension means high blood pressure. And like I said, we have our own mechanism in the body to bring the blood pressure back down when it's too high. But when those mechanisms fail, or I should say, when those mechanisms are not enough to bring it back down to normal, then the person would have hypertension. Okay, and having chronic high blood pressure is not good because it makes your heart and kidneys work harder, and that increases your risk of getting cardiovascular diseases or kidney failure, both of which are of course not a good thing. It is hereditary, and diet contributes to the risk of high blood pressure. Not just like salty food as you might think, okay, but like greasy food causes your vessels to harden, and that could cause the blood pressure to go up over time as well. Okay, let's try this out. Go ahead. Okay, thank you for participating. The correct answer is veins. Okay, the further away you're from the heart, the lower the pressure is going to be. So that is the veins. Let's try another one here. Okay, please try to participate. Okay, a lot of you are not doing the polls. Is it because you don't see the poll on your device? Is that why? Just want to understand the reason. All right. This one, the velocity of blood flow is typically the lowest in capillaries. Remember I said you have to slow down substantially in order for the gas exchange to occur, right? Think about the graph. The graph comes down, right? Okay, so it's the lowest in capillary, right? So just to recap, blood pressure is the lowest at the veins. Velocity is the slowest in the capillaries. Okay, please don't mix up those two and read the question carefully on the test. All right, here's another one. Okay, I'm just gonna wait a couple more seconds. So what affects velocity? Pressure and diameter, right? So the higher the pressure, the faster. So we want something that has high pressure. So automatically this one and this one is wrong, and we want high pressure. And then if we want it to be fast, we want large diameter or small diameter. Well, I told you, right? The bigger it is, it's like having more lanes in the highway. You can go faster. So that's why C is the correct answer. Okay, if you have a blood vessel that has high pressure in it and it's really big, then it's gonna be the fastest. Okay. Do we have time for this? Let me see. Let me see how much more we have to go. No. Okay, yeah. We can do it. Okay, this is actually in your study guide. Okay, so maybe we can switch over to the study guide over here. I think it's on the first page. Sorry, like these things are not in order because like I try to save paper. If I put everything in order, then it turns out things don't fit. Like tables might, you might have half a table on one page and then the other half on the other. So like try to make it look nice. That's why it's like all over the place. So over here, number 3. Why don't you take a moment and do that? And we'll take it up in a second, okay? In a few minutes. Okay, so please give it a try. Okay, let's take it up. Higher blood pressure, that would be the artery because it's closer to the heart. Thicker vessel wall, that would also be the artery because in order to be able to withstand the high blood pressure, it needs to have a thick wall. Otherwise, it would rupture easily, right? And we don't want that. Larger lumen diameter. Lumen is the inside, and so if you remember that picture I showed you with the artery and then the vein next to it, right? The vein is the one that has a larger lumen. Contain valves, that would be veins. That is for venous return, right? In the legs, the valves would catch the blood so it doesn't fall all the way back to the foot. Blood has higher velocity. That would be artery. Slightly higher than the veins, and carries blood away from the heart. That would be the artery as well. Okay, hopefully that makes sense. And again, if you need clarification, you can email me, you can leave a message in the chat box, whatever you feel like is better. So that's it. Okay, so for each lecture, I try to go through the anatomy, which is what do you call different things, and then the physiology is how things work, right? So like the circulation in this case, and then I usually would end with a little bit of diseases, okay, associated with that particular system. I mean, we talk about some conditions throughout the lecture as well. We talk about like pericarditis, we talk about arrhythmia, but now we're gonna focus on more details on the selected few conditions. So you probably have heard of cholesterol. Okay, we have the LDL and we have the HDL. Do you know which one is the good one? Which one is the good one here? Is it LDL is a good one or HDL is the good one? Which one is that? If you're gonna have some cholesterol, which one would you rather have? Anybody?
HDL.
HDL, yeah, HDL is the good one. Okay. I mean, you don't want to have too much of it, but you do want to have a higher HDL than LDL. You want to have a better ratio of HDL over LDL. So what is LDL? What is HDL? It turns out the cholesterol, which is a type of lipid, you know that from last semester, a lipid don't dissolve well in blood. Okay, because blood is mostly water, and so all these lipids won't be able to dissolve in the blood, right? And in order for you to transport these cholesterol in the blood, you actually have to take it to the liver first, where it is incorporated into these lipoproteins. Okay, so it makes the cholesterol soluble, basically. If you have these low density lipoproteins, then they are very high in cholesterol content, and these things tend to build up in the linings of the arteries. Whereas high density lipoproteins, they are the ones that have a lower cholesterol level, and the liver actually use it to form bile. We learn more about bile later, but bile, it's that like bitter stuff that your liver makes, right? That helps you digest fat. You know, if you ever get like severe food poisoning and then you vomit, you know, a lot, after your stomach is empty, after you vomit everything out, and then you still vomit, and sometimes it tastes bitter, right? And that's the bile that you're vomiting. It looks green, it looks yellowish. It makes you look like the person from Exorcist, right? If you don't know what I'm talking about, if you've never vomited bile before, congratulations, good for you. I don't wish you ever experienced that. But you know, if you ever had that experience, you would never forget it because it's so unpleasant. Okay, we'll learn more about bile later on in the course. Okay, bottom line, if you're going to have some cholesterol, you want to have the HDL and not so much LDL. If you have the LDL building up in your vessel, then it's going to cause it to harden. Okay, it's going to cause it to become less elastic. Okay? And the buildup of these fatty substances in the body is called atherosclerosis. Okay, is the hardening of the artery, the scarring and hardening of the artery. We call it atherosclerosis. Okay, and so let me see what's on the next slide. Yep. Okay, so that's what it looks like. But let me just explain it briefly first. Over here we have our blood vessel. Okay, it doesn't have to be a capillary. Okay, it could be like an artery. Okay, I told you last class the inside of the blood vessels should be smooth, because blood doesn't like going through a rough patch. Okay, anytime it goes through a rough patch, it's going to clot. Okay, so for example, if you have a damage, like a thumbtack goes through your blood vessels, right, that causes it to be damaged, and there's all these rough exposed tissue. Then when the blood goes through it, it will stick to it, and it will form a clot and eventually help you heal, right? So that's like a normal healing mechanism, and that's okay. We want that. Okay, but if we have too much cholesterol, too much LDL, then what happens is the cholesterol start to accumulate here. Okay, it starts to build up. Okay, so we have like a atherosclerotic plaque, so fatty substances, we would just say. Okay, and then what happens is when the blood comes, right, it will now pass through this rough patch, and it will actually stick to it. Okay, I will use a different color. How do I get more color? Okay, whatever, I'll just use purple. Okay, obviously it's not purple in the body, but it will stick to it, right? And then it will cause blood clot, the platelets. Okay, and then it will make it even more rough than before, and now you will have more things getting stuck to it, and more things getting stuck to it, and eventually you get this big blood clot. Okay, and then it makes this part of the vessel harden, makes it weaker. Right? That's what atherosclerosis is. Okay. Now, what's the problem with that? Well, couple of problems. Let's talk about the obvious one first. This part is hardened, like I said, right? Is weaker. So it's possible that if the blood going through it has like really high blood pressure for some reason, then the high pressure would just cause this part to burst. Okay, and then you would have a hemorrhage. Okay, it depending where it happens, maybe it's not as big of a deal if it happens somewhere in the body, but if it happens in the brain, then it's very serious, right? Have you guys heard of like an aneurysm? Aneurysm, anybody heard of that? Want to share what that means? So, oh yeah, go ahead, Jamal. Yeah, go ahead.
Isn't it when a vessel in your brain bursts?
Yes, basically that's what it is. Okay, so this is a vessel that is in the brain, and it's weakened, perhaps by something like this, and you know the person normally would not even know about it, and then one day maybe the person slipped and fall and then hit their head, and that blunt force to the head creates enough force for the weakened blood vessels to just burst, and then the blood would leak all over the brain, and that's what causes the aneurysm, right? And then the person could die from it. And you know, you actually might read about this on the news of some seemingly healthy person after being hit in the head, either because they fell or they bumped their head somewhere, just drop dead, right? And a lot of the times is because of aneurysm. So that's pretty scary. Another thing that could happen is let me just erase all these things coming out. Another problem is oh, someone messaged me, when you go skydiving and toy have aneurysm, does the pressure pop it? Oh yeah, the extreme sport, right? Also could place stress on these weakened vessel. Doesn't have to be skydiving, right? It could just be like, you know, going like playing like a football or something and getting hit in the head. But anything that puts stress on the body could potentially pop one of these things, and of course it's very dangerous. And the most dangerous thing is like you wouldn't know about it. Like you wouldn't know you have a weakened vessel in the brain, right? There's often not a lot of symptoms, so you could be seemingly healthy, and this just happens out of nowhere. What was I talking about before that? Right. Another thing is this blood clot is really weak. So when the blood is just rushing by, sometimes it's possible that this whole blood clot just kind of get pushed off, right? And then the blood clot would just fly off and then continue traveling into the blood vessels. And so if this thing goes on to like an arterial and then to a capillary, because capillaries are really small, right? And even the arterioles are smaller than the arteries, this blood clot can get stuck in one of the smaller vessel and completely cut off blood supply to whatever body parts is supplying to. Okay, so we call that like an embolism, and that could cause a part of the body to basically die off because it doesn't get enough oxygen supply to it. Okay, so that's another scary thing that could happen. So here is a regular blood vessel. You know, you taking the blood vessel and then you're cutting it like that, and you can see the inside is unobstructed. And over here we can see the atherosclerotic plaque. That's the fatty substances that's blocking the vessel. But the majority of the vessel is actually blocked by the blood clot, right? The blood starts sticking to it because it was a rough patch, right? And then it's just kind of like a snowball effect. It just gets like more and more severe until the vessel is really small. Okay.
Professor, is that what would cause a stroke?
Yes, that is exactly my next line. Thank you for bringing that up. But before stroke, we will talk about heart attack as well. Okay, so coronary circulation. At the beginning of the lecture, not today's lecture, but like last lecture, I told you that the heart cannot use the oxygen in its chambers. Okay, the blood is passing through the ventricles and the atrium at really high speed and really high pressure. It's impossible to extract the oxygen from those blood. And in order to get oxygen for your heart muscle, for your myocardium, it relies on these coronary arteries. Okay, coronary arteries. So on the surface of the heart, right, you can see all these blood vessels. In this particular specimen, they dissolve all the muscle and maintain the blood vessels, and you can see it's an extensive network of arteries, right? That's continuously supply oxygen to your heart muscles. Now, hold on, I'm just gonna see what to say next. Okay, so if you have one of these coronary vessels being blocked by the blood clot, for example, or by the atherosclerosis, if it's partially blocked, then what happens is it could lead to what we call coronary artery disease, or CAD. Okay, so in this case, it's not completely blocked, it's just like a little bit blocked. So that means you're still getting blood supply to your heart muscle, and it might be okay if you just not doing much. However, if you physically exert yourself, let's say you walked up a flight of stairs, you try to lift something heavy, then all of a sudden your heart muscle it demands more oxygen, then the partially blocked vessel might not be able to supply the extra oxygen needed. Okay, so people who have a partially blocked coronary artery, when they physically exert themselves, they might have chest pain, and that chest pain, we call it angina. Okay, so like you hear about people warning about shoveling snow in the winter, right? Like make sure you don't overexert yourself, because like you don't know if you have a heart condition. You don't know if some of your coronary arteries might be partially blocked, right? And when it's really cold, your blood vessels actually shrinks, right? Vasoconstriction, and that could make the situation worse. Like if you already have a slightly blocked artery, when it's really cold, it might make it even more blocked because of the vasoconstriction, right? And so people shoveling snow, they might suddenly experience chest pain, the angina. Okay, now obviously, if you experience chest pain of any sort, you should get it checked out. And they could do like an angiogram. I don't know if you heard about that. They basically put some dye in your vessels, and then they do a chest X-ray for you, and you will be able to see which arteries are blocked or partially blocked. And if they determine you have a partially blocked coronary artery, the doctor might prescribe you some aspirin. Okay, and aspirin is basically a blood thinner. Okay, it makes your blood less viscous, makes it less thick, and so it's able to pass through narrowed vessels easier. So people who have coronary artery diseases, they might have to pop an aspirin, on a daily basis. If you go to the dentist and you fill out that questionnaire, sometimes they would ask you. Well, actually, they always ask you if you take aspirin as a daily medication. That's because as you know, when you clean your teeth, right, there's a lot of blood, right? And if you're on a blood thinner, it might cause your blood to not clot as quickly as possible, and then they need to know about it. Okay, so that's aspirin. Another one is nitroglycerin. Okay, nitroglycerin. You might have heard of nitroglycerin is actually a potent explosive. It blows things up. It's being used, you know, as the weapons grade of it is used as explosive to blow things up. But I don't know who discovered this. But if you put it in a little bit of it into your system, into your body, it actually would cause your blood vessels to dilate. Okay, so people can either put the pill underneath their tongue. You might have heard about that when they experience the angina, and that would cause the blood vessels to open up and alleviate the pain. Or sometimes there is the spray, okay, as well, and that would again cause the blood vessels to open up and restore the blood flow. All right. Neither of these are like permanent solutions. Okay, they're just like band-aid solutions. They are just treating the symptoms. They doesn't really address the cause of the problem. The cause of the problem being that your blood vessels is, you know, partially blocked, right? And so sometimes people can do what's called balloon angioplasty. Okay? This involves inserting a catheter. Usually they do it up from the legs, from the femoral artery, and then they feed it up to the heart, where the location of the blockade is. And of course, this whole thing is guided by ultrasound, right? So the surgeon knows exactly where to go. And the tip of the balloon is an inflatable. Sorry, the tip of the catheter is an inflatable balloon. So after you go to the blocked vessel, let me try to draw this out for you. There is your block vessel, and then now you have this balloon that's coming in. Then you inflate a balloon. You inflate a balloon. So after you inflate a balloon, the balloon gets big, and then it will flatten the plaque. And then you do that a couple times, and then you remove the catheter. So afterwards, your blood vessel would have a flattened plaque, and you would you know, restore the blood flow. Okay. But about, you know, 20 to 30% of the people who undergo this procedure will experience restenosis, okay, re-narrowing of the blood vessels, which makes sense because you're not getting rid of the rough patch, so it's possible for more blood clot to form later, and so, you know, you might have to go back and do another procedure after some time. Does that make sense? Now, this is not like without risk. It's possible that, you know, when you open up the balloon, some of the fragments might break off, and then this gets caught in some smaller vessel that could cause problem. And to prevent the restenosis, sometimes they actually put a stent in there. Okay, they put a little metal stand that props the veins open, so it's not possible for it to reclose again. But you know, you have to make sure the stent is made up of a material that is not going to trigger your immune system, because otherwise that's just going to cause more clotting to occur, right? Okay, so that is that. So in other cases, if you have like a complete blockade, we were talking about partial blockade, right? If you have a complete blockade, then it could lead to a heart attack. And the fancy word for heart attack is myocardial infarction. Infarction means to die. So your myocardium, your heart muscle, died as a result of not getting enough oxygen. If you survive the heart attack, then they might do what's called coronary bypass surgery for you. Is it written here somewhere? I can't see. I'll just write it out. Coronary bypass. So in coronary bypass, you would take a vessel from your leg. Apparently, you have a lot of overlapping veins in the leg that you can take some out without affecting its function. And then they would connect the vessel directly from the aorta to downstream of where the block occurs. So let's say there's the block here. So the blood come down here, and it's not going to be able to get through. So this entire area would have no oxygen supply, right? By connecting a vein directly from the aorta to behind the block, you will now create a detour that would supply oxygen. Okay, so this is This is coronary bypass to go through the clot, basically. So earlier, someone mentioned about stroke. It's essentially the same mechanism here, except rather than happening in the heart, this occurs in one of the vessels in the brain, and depending on which part of the brain is being affected, the outcome could be severe or it could be mild. So that is all for lecture 1, and you will be quizzed on this in week number 3. All right. Now we still have some time, so we will fill in more of our study guide here. I don't know if you guys had a chance to go through it, but in case you did, any particular requests for these questions? Is there like a particular section that you really want me to take up? Anybody? Okay, I guess we'll go through the true or false here. First question. The tricuspid and the bicuspid valves are both known as atrial ventricular valve. Okay, so think about whether that's true or false. You can just quite quickly type me a T or F in the chat. Okay, so I know what you guys are thinking. Okay, I'm getting a couple of answers. Just waiting for more people. Tricuspid, bicuspid. Are they the AV valves? Yep, this one is true. Okay, very good. Next one. The pulmonary arteries take oxygen-rich blood from the heart to the capillaries in the lungs. Is this true or false? Okay, I'm getting a lot of falses. Some people is answering true. Okay, yeah, this one is false. Okay, pulmonary arteries take oxygen-poor blood from the heart and take it to the capillaries. That's the reason we take it to the lungs, right? If it's oxygen-rich, we don't need to reoxygenate it, so that's why it's false. A stroke occurs when one or more of the coronary arteries are blocked. We just talked about this. This is false. Coronary arteries, if you block it, you will get a heart attack. Fancy word for heart attack is MI, myocardial infarction. What about the next one? The AV node is also known as the pacemaker of the heart. True or false? Let me know. Okay, I'm getting mostly false. That is correct. It is not the AV node, is it? It's supposed to be the SA node, the sinoatrial node, SA node. The systolic pressure is the pressure in blood vessel when the ventricles relax. That is also false. Contract, right? If it's relaxing, it should be called diastolic pressure. Osmotic pressure will increase if blood sodium level drops. Let's think about that one. What do you think? True or false? Osmotic pressure will increase if blood sodium level drops. Okay, so let's talk about this one a little bit more. This is your blood vessel. And then, let's say it's a capillary. And we know that the osmotic pressure is dependent on the solute concentration, right? Okay, so this is solute, this is the blood concentration, and then this is concentration on the outside. So normally you would have higher blood concentration compared to the outside, and that's what's creating the osmotic pressure. Okay, so what happens if the blood sodium level drops? Okay. If you have lower blood sodium, then you will have less dots on the inside, right? Fewer dots. Okay. So now the difference is not as drastic, then you won't be bringing in as much fluid. I mean, you will still be bringing in some, but not as much. So if you drop the sodium level, the osmotic pressure will actually decrease. Okay, so that's false. That's why people who have hypertension should cut down on their sodium level, right? Because if you increase the sodium level, you will have more dots over here, and you will end up drawing more fluid into your blood, and that could increase your blood pressure even further. Okay, so that's that. So a lot of the multi-select questions on the quiz or test, they are basically a combination of these true or false, right? It would say choose all the false statements below, right? Or choose all the correct statement below. Okay, so you have to really think about it. Sketch the 3 graphs. I'll let you do that on your own. I will post the answer for the fill in the blanks as well. And I just want to end the lecture by talking about the application questions. Okay, let's take a look at this. So by the way, the application question that I'm talking about here, I could convert this into a application multiple choice on the quiz, right? So that's why you should pay attention here. All right, first question. Congestive heart failure occurs when the ventricles fail to empty its content completely. Speculate what happens to someone if congestive heart failure occurs on the right side of the heart and then on the left side of the heart. Okay, so if you get a question like this on the test, and then you will have different choices, you might be like, we never talk about congestive heart failure in the class. How am I supposed to answer it? But then it doesn't really matter because the question actually defines congestive heart failure for you, right? It's a condition where you don't empty completely. And then your job is to think about, oh, if the chambers are not emptying completely, what's the implication of that? That's why it's an application question, right? You take what you've learned and apply it to a situation like this. So I will explain this to you. You know what? I'll just explain it in the notes right here. Okay. So congestive heart failure. I'm just going to draw the typical heart, and then we have 4 chambers here. Okay. Let's talk about the right side, right congestive heart failure. Right. So the question was saying, it happens when the ventricles is not emptying completely. So on the right side, right, if the ventricle, this is the ventricle, right? Maybe I should label it. This is right atrium, this is right ventricle. So let me choose the right color here. When the right ventricle squeezes, right, all the blood is supposed to go to the pulmonary arteries. But if we have congestive heart failure, then we're going to have some leftover blood that is just hanging around. It's not able to exit. Okay. If you have leftover blood from the ventricles, then next time when the atrium tries to empty and fill up the ventricles, because of a backlog here, now it's not able to empty completely either. So it will also have some leftover blood. Right? It's like traffic jam, right? It just creates a backlog. And so what vessel feeds into the right atrium? We have the superior vena cava, we have the inferior vena cava, and these things comes from the body, right? So if you think about the body right here. Okay. Now, because the right atrium is not completely empty, all these blood coming from the body is not gonna be fully returning to the heart as well, and so you might have extra blood pooling in the lower half of the body. Okay? So superior vena cava is probably okay. You're not going to have blood pooling in the brain because gravity will take care of that, right? But you will have some blood pooling at the bottom. And if you zoom in to the blood vessels in the leg, right, that extra bit of blood will actually cause increase in blood pressure, which will push more fluid out. So you will have increased fluid loss. Okay, so people who have right side congestive heart failure, they usually would have swelling in legs. Okay, swelling in the body in general, but particularly severe in the legs. Do you understand? Okay, so that's the thought process that you have to go through. Now, you're not going to have like too many of these application questions. There's just going to be a few, and they're only 1 mark each, okay? But you know, that's how you would approach something like that. So using the same logic, does anybody want to speculate what happens for left side congestive heart failure? Left congestive heart failure. How's that going to affect the body?
Poor circulation.
Sorry? I didn't catch that. I'm sorry.
Like poor circulation to your extremities.
Okay, poor circulation to extremity. Yeah, that could be it, because you're not emptying completely, so you might not be getting as much blood to your systemic system. Zia said the blood in the left area will not be enough to supply the. Yeah, okay. So basically, what Min was saying. What about like swelling? Do we expect any swelling? If so, where? Okay. So what's supplying blood to the left side? What's supplying blood to the left side? What vessel is this? Right, this is the pulmonary vein, right? So if you have a backlog of blood going on here, then the pulmonary vein is not going to be able to come to empty completely, and that causes buildup of fluid in the lungs. So for left congestive heart failure, you would have pulmonary edema. Okay, edema is swelling. Okay, fluid accumulation. Okay, so instead of accumulating in the legs, you would have accumulation in the lungs. All right. So for people who have congestive heart failure, it could be congenital. People who are born with it. It could be because they're recovering from heart attack and their muscle is weaker, so it's not able to empty completely. Right. So they would need to take some drugs. They're called like cardiotonic that strengthen the heart muscle, and that could correct the problem. All right. Let's take a look at the next one.
I have a question.
Yeah, please go ahead.
So with the pooled blood that's left in the heart, does it harden or clot? Does it?
Oh yeah, no, not really. So when the next set of blood come, it kind of mixes the old blood, right? And then it would get sent off through the next cycle. But you will always have some that's left over. But it's not like the same blood, right? It's just like, you know, it's like blood. So let me see if I can explain this better. Okay, so let's say this is
So it's like a blood reservoir that you don't want.
Yeah, exactly. So there is some blood left over, and then when new blood come, right? It's not like only the new blood will get sent away next time, right? Like the new blood will mix with the purple blood, and then it creates like a new mixture. But then next time that you send it off, right? You will have like another new batch that are. I don't. Does that make sense? I'm not really explaining this too well, but it's basically you always have some left behind. Think of like your cup of water. Okay, you have some water in there. Okay, you pour it out. You have some left, and then you add more water to it, and then you pour it out, and you have some left over. You always have some left over, but it's not necessarily like the same water all the time. It's not the same blood that's pooling all the time. They don't get
So
clogged because the pressure will keep on washing them off, right? Sorry, go ahead, Dashana.
So basically, Professor, it does get pushed out to the body, but not with the batch that it comes in with.
Exactly, right? And you the key thing is you're not pushing all of it out. So instead of supplying your body with 100% of the blood, because of this issue, you may be only supplying it with like, I don't know, like 90% of the blood. Do you understand what I mean?
So then the real problem is not the actual blood itself, but it's just like a weak pumping heart.
Exactly. Yeah. It's basically the ventricles, right? And the heart is weakened. Like I said, either is the person is born with this problem, right? Or they are recovering from a heart attack. Okay, heart attack causes muscle to die, and now it's weaker, it's not able to empty as much. Okay.
So then what's the solution to this problem?
Right. So earlier I was
Okay.
saying, right, they could take a drugs, medications called cardiotonic, TONIC, that would strengthen the heart muscle. Okay, it causes the heart to squeeze better, to increase, to improve the stroke volume, basically, and that causes to empty. Okay, that's one of the solutions.
Okay, thank you.
Yeah, very good questions. All right, one more, and then I'm gonna let you go. You have been sitting here for 2.5 hours, so thank you for your attention. A heart block occurs when there is an interruption in the conduction system of the heart. Okay, the conduction system, as you might remember, is the SA node going to the AV node and then going to the Purkinje, right? Purkinje fiber. In first degree heart block, right, the conduction from SA node to AV node is delayed. All right. So based on this definition, a heart block is an interruption of the conduction system. It's not a physical block. It's not like you have something stuck in your vessel and blocking it. Okay. A heart block is when the conduction system is interrupted. So rather than going from SA node to AV node immediately, it is delayed. What changes would be expected on the ECG of someone with first degree heart block? This is a normal ECG. So how would it change, right? So, do you have a higher P peak? Do you have a longer a period between, Q and S, or like smaller R? What do you think? What are some possible changes in the ECG that you would notice for someone who has a first degree heart block?
The P wave will be longer.
Okay. Yeah. Anyone else? Thank you, Shelley. So she said the P will be longer. Anyone else? Low QRS. Okay. So like a lower spike here. I think that's what you mean. Anyone else?
The spikes will be further apart.
The spikes will be further apart. Okay, so all these are excellent suggestions. Let's take a look at our PowerPoint slides again. If I can find the ECG, that would be okay. So over here, right? So hopefully you wrote down the notes and you can flip back to it and for your reference. Unfortunately, I lost everything I wrote already every time I exit. But the first hump here, right, is corresponding to the atrial contraction, and then the spike is corresponding to the ventricular contraction, and in between, that's when the signal is going down to the ventricles, right? That's when the signal is spreading. So if you have a delay, in conduction between SA node and AV node, this part here will get longer. Right, you will have the atria contracting, but because the signal takes some time to get to the ventricles, this would have a longer delay before you get that spike. Okay, so longer P to Q interval. Does that make sense? I mean, this is an exaggeration. It's not going to be like super drastic long. Like you would probably die if it's really, really long. But there's going to be a delay. Does that make sense? So that's some examples of application questions. Hopefully it gives you an idea of what to expect. And again, I'm telling you, like, I'm not trying to make it impossible for you for the test. It's very doable. As long as you study and go through the notes, you should do reasonably well. Okay, I have 20% of the class last semester doing this online, 20% of the class gets higher than 90% in the course. Okay, so it's very, very doable. All right. So thank you for coming. That's it for this week. I will see you next week for lecture 2, which is blood. It's a very fun lecture. We talk about heredity and figuring out the blood type of the kids, and what happens if the mother and the child has blood incompatibility. What's the implication of something like that? So I'll see you all next week. Okay, Monday 8:30. Okay, have a good one. Bye.